Hello. In this unit, we're going to be talking about diversity of life, and in fact, throughout much of this third semester, and third quarter into the fourth quarter. So this is going to specifically cover chapters 26 and 27 from the text, in which 26 we study phylogeny, or what I should say is the science of systematics, and that is the organization of life, the relationships between living things in which we develop these phylogenies, or these trees. Again, systematics is the way we do this. That's the science of classifying things, the relationships between different species. There are many ways to do that. Traditionally, you used morphological data, just how things look, to classify them, but more so nowadays we're also using biochemical and molecular data to figure out relationships, that is, similarities between the DNA of different organisms to study their relationships. From these data, you of course generate trees, if you will, phylogenetic trees. That's how Darwin envisioned it, like a tree in which the trunk was the sort of the first living things, and the branches that come off of that trunk are the major groups of organisms, and then the tips of particular branches are individual species. And again, nowadays we use both morphological and molecular data to figure out these trees. So, a particular morphological trait that is of a curious nature, what are called homologous traits, or homologies. And you can see here we've got the forelimb of 4 different types of mammals. And they have the same kind of bones in them, basically, but they're just different arrangement of them, shape of them, size of them, if you will. And so the fact that they have the same bones strongly suggests that these 4 organisms have these 4 species have a common ancestor. But each of them uses the trait, this forelimb, for a different purpose, because through evolutionary time it's been adapted and changed towards what that organism does, whether it grabs things, walks, swims, or flies. So again, a homologous trait is one that shares a common ancestry but is used for different purposes, whereas analogous structures are ones that are the result of convergent evolution. They're species that don't really share a common ancestor, at least not one in the near evolutionary past, but they have characteristics that are the same because they've evolved those characteristics because they live in a similar habitat. So one of these represents a marsupial on the top, and the other is a shrew, a placental animal. They're both digging animals, but again, they don't really have a close common ancestor at all. Molecular homologies. Well, when we look at molecular data, again, we're looking at similarities between them. We can look directly at the DNA and look at similarities between sequences. And so you can see these 2 organisms share a bit of similarity, which would suggest they have some kind of common ancestry. You can also look at protein data. You can look at amino acid sequence and proteins to figure out similarities between species. And so all these organisms may have one particular protein they share in common, but if you looked at the mouse and the chimp, theirs would probably be much more similar than, say, the chimp versus the salamander. Cladogram. A cladogram is basically a phylogenetic tree that shows these patterns of characteristics that organisms share. And so when you have these cladograms, what you do is you label them with particular characteristics that we can see here. Jaws. All these organisms from the perch on up all have a jaw, whereas the hagfish does not. Alright, now what's next? The lung. All these organisms from the salamander up have lungs, claws or nails, and you can see feathers specifically in the bird, and with the mammals, fur and mammary glands. So essentially, none of these guys have fur or mammary glands, but everyone else on this tree does. So a cladogram, again, shows a particular trait and where it falls in the tree and which organisms have that trait. You can speak of primitive and derived characteristics. So primitive characteristics are ones that go way back in evolutionary time and are shared by lots of organisms, whereas derived ones would be relatively speaking, would be a more recently or, again, derived characteristic. So the jaw, for example, is a derived characteristic relative to the hagfish. fur is a derived characteristic in these organisms relative to these other species. All right, so people who study systematics have attempted to classify organisms for centuries now, if not thousands of years. And so you may remember studying freshman year, we often classify things into the 5 main kingdoms. That is, we just say bacteria and then protists, plants, fungi, and animals. Well, another common way of doing this is dividing life into 3 domains: the archaea, the bacteria, and the eukaryotes. Basically, you can see the prokaryotes are split into 2 separate groups here, or 2 separate domains, whereas the eukaryotes are all in the same domain. In this slideshow, we're just going to focus on the prokaryotes. We'll get to the eukaryotes later. Now, one thing you want to keep in mind is sometimes we may think of these of different species, different groups of organisms, different domains for that matter, being relatively isolated from each other. But in fact, there is a fair amount of movement, particularly of genes between these groups. You know, within the prokaryotes, there's been documented many examples of horizontal gene movements between what we would consider separate groups or separate species. And of course, early in evolutionary history, we had the movement of structures from the prokaryotic domain into the eukaryotic domain, essentially forming the eukaryotic domain via the addition of mitochondria and plastids. And of course, these guys brought some DNA with them. They have their own genes. So essentially, you have primitive prokaryotic genes inside of your mitochondria, for example. Okay, so that's just a general look at diversity. Now let's focus on prokaryotes. So you've learned about them in the past. We know they're single-celled organisms, relatively simple. Some of them live by themselves, some form colonies. And they come in these 3 basic shapes: the coccus shape, the bacillus, and the spirillum. And again, you can see sometimes they'll form colonies or clusters of cells. Okay, gram-positive bacteria. These are bacteria that have a particular cell wall and membrane structure. They have a really thick cell wall, and the cell walls of bacteria are made of, or prokaryotes I should say, are made of this polysaccharide called peptidoglycan, which you don't see in the cell walls of plants or fungi. So in that sense, cell walls are an analogous trait. There are many organisms that have them, but they didn't evolve from a common ancestor. So anyways, gram-positive bacteria have this really thick cell wall that retains the stain that makes them dark purple, and they're known as the gram-positives. Others that have a thinner cell wall don't retain the stain, and they show up as sort of pinkish color, and they're known as the gram-negatives. Gram-negatives often have a double membrane, whereas the gram-positives have a single membrane. Here's the wall right here. And when you watch this slideshow, you can click on this link to look at some gram-positive bacteria. Motility. Some prokaryotes are able to swim. They have a flagellum. Now, this is also another good example of an analogous structure, because we have eukaryotes with flagella, but the structure of the flagella is completely different. In eukaryotes, it's an extension of the cell membrane, but in bacteria and prokaryotes, that's not. It's a protein structure that basically sticks out of the cell membrane. Does the same thing, but again, it's made of it has a different structure and is therefore analogous. You can click on this link to see a video of some swimming bacteria. Now, we do say that prokaryotes in general have a simpler internal structure. They don't have a nucleus, they don't have mitochondria, they don't have chloroplasts, they don't have endoplasmic reticulum, they don't have Golgi apparatus. They do have ribosomes, but some of them have developed sort of a more complex internal structure. And so they've taken the outer membrane and folded it into a complicated inner membrane. And in particular, you have certain bacteria that are really good at carrying out cellular respiration. And so they use that inner membrane as the site on which that happens. And the same with photosynthetic bacteria. They don't have chloroplasts, but they've taken the outer membrane and basically folded up a portion of it inside them to provide the surface area for the light reactions to occur. All right. Bacteria, again, they primarily reproduce asexually, but they have the ability to acquire new genetic material. And we talked about this a little bit in genetics via conjugation, where they can get together and form a little connection between them, and then DNA via a plasmid is transferred from one to the other. You're aware of transformation as well, whereas the DNA from a bacterium that is dead, that is broken apart, can be taken up by a living one and become part of its genome. And then also transduction, where you have a vector, typically a virus, that moves DNA from one bacterial cell to the next. The virus picks up bacterial cell when it's sort of breaking out of the one host bacterium. You remember viruses typically want to, of course, carry their viral DNA, but occasionally a virus capsid will pick up bacterial DNA instead, in which case when it attaches and injects that DNA, it's not taking over that host to make it into a virus-making machine, but it's inserting bacterial DNA. All right, the ecology of prokaryotes is a big deal. We're going to spend a good chunk of the rest of the slideshow talking about that. They come in many forms, many nutritional modes. You have some that can make their own food, some autotrophs, and some that can't, the heterotrophs. Some of these autotrophs are what we call photoautotrophs. They use light as a source of energy. They're basically doing photosynthesis the same way as plants. But then there are others that use chemicals as a source of energy instead of sunlight. And I imagine you can think of where you might find some of these chemoautotrophs, basically where there's no light. So you'll find them in the sediments of swamps and in aquatic systems where it's deep enough that you have little or no light. So they make use of a different source of energy to turn carbon dioxide into organic compounds. Heterotrophs, we are heterotrophs. We can't make our own food. There are bacteria that are heterotrophs, and many of those are chemoheterotrophs. They use the organic compounds they consume as both a source of energy and raw material to make more of themselves, and that's what we do. But then there are some that are curious. They take in organic compounds as raw materials, but they use sunlight as a source of energy to drive building those organic compounds into more complex things. A rather unusual way of doing things. Oxygen. Of course, we learned a while back about how when photosynthesis evolved, there were photosynthetic bacteria that increased the oxygen in the atmosphere, and so that changed life for many organisms, turning them into aerobes. And you have some bacteria that are what we would call obligate aerobes. They have to have oxygen in order to survive. Some, though, do not like having oxygen around. They're the obligate anaerobes, and they are poisoned by oxygen. And because there is so much oxygen in our atmosphere, you tend to find those in particularly unique environments, again, say in the sediments or swamps, and they also live in the guts of certain animals. And then you have the facultative anaerobes, and these are ones that can survive with or without oxygen. E. coli is a good example. It doesn't matter whether oxygen is around, it'll keep doing its thing. Okay, I'm running out of time, so I'm going to stop here and do a second video to finish out our slideshow.